All right, Dr. Kathy Neto is Associate Professor in the Department of Chemistry and Biochemistry at UMass Dartmouth and a Director of the newly formed UMass Cranberry Health Research Center. She earned her PhD in chemistry from Brown University. Following postdoctoral study on antiretroviral agents, she joined the faculty at UMass Dartmouth and developed a research program on the chemistry, biological activities, and potential health benefits of natural products from cranberries and other fruits. Dr. Neto has authored several review articles on cranberry phytochemicals and health, and has been invited to speak on this topic at national and international meetings. She has collaborated with sciences throughout UMass, the University of Prince Edward Island, WPEI, Cornell, and Bruker Biospin, supported by funding from the Cranberry Institute, the Wisconsin Cranberry Board, the NIH, the USDA, and Canadian funding agencies. Let's welcome Kathy Neto.
Thank you. Can everyone hear me okay? Okay. I just want to say thank you for the delicious cranberry bread. It was outstanding. Whoever made that, really, really good, and good for you. So what I'm going to attempt to do is to talk about how the role organic chemistry plays in a very multidisciplinary field of functional food science. So I'm going to be talking about, in general, berry phytochemicals, but specifically about compounds in cranberry and some of their mechanisms of action, some of their organic chemical structures, and why we're interested in them. So you are what you eat. So if you're eating cranberries, you're off to a good start. If you're eating blueberries and kale and all those other good things too, even better. So what I'd like to try and do is to introduce some dietary phytochemicals. And phytochemical is a term that refers to compounds from plants. So they're natural products, but specifically from plant sources. And talk about how some of these may play a protective role as the body ages. So I'll be talking about some of the science behind functional foods, which is the way we in the industry describe foods that contain compounds that have potential biological effects. And then I'll be talking more specifically about some of the compounds in cranberries and how we study their structure and their properties. So if you're wondering what organic chemistry has to do with all of this, first of all, phytochemicals are a branch of natural products, and they're organic molecules that are produced by plants. We usually refer to them as secondary metabolites. So Susan had introduced some of the basic molecules that are necessary for life, and some of those, like carbohydrates, proteins, etc., are what we call primary metabolites. So they're necessary for organisms to survive. Secondary metabolites are usually produced by organisms in response to some stimuli. So it might be a need for defense against some attacker or just environmental stress in general. Now, cranberry and other phytochemicals are organic molecules and primarily contain carbon, hydrogen, oxygen, and nitrogen. So there is a whole branch of natural compounds from plants called alkaloids that we were talking about earlier, and those are nitrogen-containing compounds. When we talk about functional foods, most of the compounds that we talk about are not alkaloids. Alkaloids do have strong toxicities, and some have psychological, psychotropic effects, and so forth and so on. So they play a strong medicinal role, but from a nutritional point of view, we focus more on compounds that are mainly carbon, hydrogen, and oxygen. So the biosynthetic pathways that lead to the production of these molecules in plants are a series of organic reactions. And some of them are the very same reactions that students learn about in sophomore level organic chemistry. I also teach a course, graduate course on natural products, and we teach biosynthetic pathways, and everybody has to think back to when they learned about their basic organic reactions, Claisen condensations, Mannich reactions, and all sorts of reactions like the ones that Susan talked about, all play a role in how the plant puts these molecules together. So I'm going to talk about two broad groups of compounds. First of all, the polyphenolics. So Susan talked about resveratrol. That's an example of a polyphenolic compound. Polyphenolics tend to be water soluble, and they tend to undergo redox chemistry, so oxidation reduction reactions, pretty readily. As such, they're considered to be antioxidants, although that term is not quite as simple as it sounds. I'm also going to talk a little bit about another group of compounds called isoprenoids, and these are the more lipid-soluble molecules. And Susan talked about lycopene, and lycopene is an example of an isoprenoid compound. It's lipid-soluble, it has a long chain with repeating double bonds. Some of the isoprenoids have that type of structure, some of them have more of a multicyclic structure. But they're not aromatic and they're not phenols. So coming back to what happens to your body as you age, well, it takes its toll in many ways, and I hate to just tell you this, folks, but it's chemistry. So yeah, chemistry is great, but not always. So your body's constantly exposed to pathogens and environmental stresses, and the stress of the job and the family and everything else, right? So that can oftentimes lead to what we call oxidative stress in the body, and this is where the antioxidants come in. And inflammation, which kind of goes hand in hand with oxidative stress. So these things are not good for the body, and they're cumulative effects. So things start to break down, your joints start to break down, and your collagen is breaking down and stiffening up, and this is unfortunate organic chemistry. It's a fact of life. So what can we do to prevent this, right? This can lead to various diseases, or just simply the aging process. So cardiovascular disease, cancers, stroke, dementia, et cetera, tend to increase as we age because of the toll that this aging process takes. So what can we do about this? Well, you can take your vitamins. I'm an advocate of vitamins. Unfortunately, a lot of the clinical studies on vitamins are not conclusive in terms of their benefits. However, some of them do support the use of vitamins in safe amounts, I should say. I'd never advocate taking too many vitamins. And vitamins are necessary. It's important for you to have a certain level of vitamins in your diet, and if you don't have it in your diet, then maybe you do need to supplement. These compounds are cofactors in important biochemical reactions, and therefore they're very necessary. But if you think about what you really take in on a daily basis, it's not going to be vitamin pills per se. It's going to be fruits, vegetables, fiber, meat, dairy, etc. So if you're eating plant-based foods, fruits and vegetables, you're going to be getting a lot of phytochemicals. So phytochemicals is just a general term, as I said, for bioactive chemical compounds found in plants. And so these are essentially organic molecules, and they contain the same functional groups that Susan talked about. A lot of them are alcohols or phenols, which is an aromatic alcohol, or ethers, which has an oxygen, carbon-oxygen, carbon bond, or ketones. And I'll be showing you some examples. Here's a compound right here. It's a polyphenolic compound. And this ring up at the top here is a phenol ring. It actually is a diphenol, orthodiphenol. And it has a ketone group too. So as Susan mentioned, a lot of natural molecules have multiple functional groups. As such, you might imagine that they have multiple effects on the body, and that's true. So a lot of our oxygenated functional groups, not only these, but also carboxylic acids, esters, are important in phytochemistry. So phytochemicals in general, many of them do possess antioxidant and anti-inflammatory properties that can help combat the effects of stress and aging on the human body. Some of these compounds, studies show, are able to protect specific tissues by the way they bioaccumulate in certain tissues in the body. For example, you may have heard that blueberries are good for your brain, and there are compounds in blueberries that are antioxidant compounds called anthocyanins, and those are able to bioaccumulate in the brain and do a lot of their good work there in the brain tissue. And much of the research on blueberries in the brain has been done right here at Tufts University by Dr. Jim Joseph and some of his colleagues. We also have compounds like lutein, which are good for the eye. They help to protect against macular degeneration. You find a lot of lutein in green leafy vegetables like kale, spinach, and you also find it in cranberries, as it turns out. You may have heard that chocolate is good for your heart. Chocolate contains compounds called catechins, which are also antioxidants, and they're thought to help support and keep your your veins and arteries healthy, so they may have a cardioprotective effect. So chocolate doesn't only just taste good, it may be keeping you alive longer. So some age-associated ailments that may be improved by dietary factors. Some of these are maybe more well-supported than others. Cardiovascular disease and stroke, a lot of research over the past 20 years does support a role of dietary phytochemicals in maintaining a healthy cardiovascular system and a healthy brain. Certain cancers, not all cancers, but some cancers may benefit from certain dietary phytochemicals in the diet. This is particularly important in the colon, where these compounds do tend to accumulate, right? When you digest your food, most of the compounds make their way to the colon. They may be metabolized along the way or metabolized once they get there, but they also may elicit some protective effects once they're in those tissues. Macular degeneration, lutein, there's a connection between carotenoids and those type of compounds and preserving your eye health. Dementia, it's thought that, for example, blueberries help to preserve cognitive function and other antioxidant sources. Urinary tract infections, which I bring up because that's one of the first areas of health research that people related to cranberries and intake of cranberries, was that cranberry juice was thought to protect against urinary tract infection. And in fact, there are some clinical studies that do support this role for cranberry juice. Now, you may have heard that red wine is good for you, and I think Susan said this too. And people haven't always known this, but there was a sort of phenomenon that was observed in France called the French paradox. And in certain regions of France where people eat lots of very fatty, high cholesterol, high saturated fat foods, they still don't get a lot of heart disease, right? So there's a connection usually between high fat intake, saturated fats, and heart disease, but not so much in France. And so studies began to show that this may be due to their high intake, relatively high intake, of red wine. And red wine, as Susan pointed out, contains compounds like resveratrol, which is a polyphenolic, and flavonoids. And flavonoids are another whole group of polyphenolics. And they're also found in berries, so I'm going to talk about flavonoids quite a bit. And so flavonoids, epidemiological studies show that flavonoids may in fact have cardiovascular benefits. One of the studies that's most often pointed to is the Zutphen elderly study, in which a large cohort of men were examined for about 15 years in terms of what they ate and their incidence of cardiovascular events. And a high dietary intake of flavonoids was found to correlate with a reduced incidence of stroke and cardiovascular events. So flavonoids are thought to be beneficial for the cardiovascular system. So how do these compounds get in the plant? Well, there are all sorts of biosynthetic pathways that lead to the production of these compounds. The shikimate pathway is one of the major ones. And why would a plant produce these compounds in the first place? Well, usually they're produced by the plant in defense. So flavonoids are antioxidant compounds, and as such, they may relieve some of the oxidative stress in the plant that the plant experiences due to environmental factors. They are also known to ward off herbivores or other predators. So the compounds are produced by the plant in defense. Fortunately for us, when we consume these compounds, they have beneficial effects towards us, for the most part. So looking at some of the organic structures here, these are some of the major phytochemicals in cranberries. And so you can see the structural diversity. These compounds are all very different. And hopefully you recognize some of the functional groups that Susan talked about. The group on the top, so those three compounds in the top row are flavonoids or polyphenolics. So those two terms are often used kind of interchangeably. Polyphenolic is kind of a broader category of compounds. Flavonoids are specifically these types of compounds with that, tricyclic structure where you have the phenolic groups here, and there are many of them. So if we take a flavanol, for example, we have multiple phenolic functionalities on both sides of the molecule. And so we have a polyphenolic. And some of these polyphenolics get pretty complex in structure. The group on the right are particularly challenging to work with. We call them packs. There's a lot of structural variety within this group of compounds. The structure of these units, so the three rings here make a catechin unit. And the structures of the catechin units may vary somewhat between different sources. So chocolate has one type of isomer. Cranberries contains another type of isomer. Tea contains some of these units with another functionality on this oxygen here. So these structures do vary, and then the number of units and the way they're linked together also varies. So plants just throw these things together, or so it seems they just throw them together, and there's lots of different ones found in berries. So it does make it a challenging group of compounds to study. Now, down the bottom, we have our lipid-soluble molecules. What you ought to notice here from an organic chemistry point of view is that the compounds on the bottom have a lot less oxygen, a lot less OH groups, so they're a lot less likely to be water-soluble. They're a lot less likely to participate in hydrogen bonding. They do, however, have a lot of lipophilic or repeating carbon-carbon bonds, a lot of hydrocarbon in the structure, which means that they tend to associate with lipids well. So this group of compounds is called the isoprenoids, and they are built by assembling little 5-carbon units. So your plant will assemble these little 5-carbon isoprene units together in different arrangements, some of which are mainly linear, some of which contain multiple rings. This is one of my favorite compounds. It's called ursolic acid. It's a triterpenoid compound, which basically means that it has 30 carbons. And it has a very steroid-like structure, at least in part of the molecule. And we think that some of its biological properties are due to that similarity with steroids. So coming back to sort of on a more molecular level, what's happening in the body as we age. So oxidative stress is one of the #1 major problems, and it's associated with many different diseases: cardiovascular disease, diabetes, cancers. In those conditions, there's an elevated level of oxidative stress in our body, and it may be chronic, and so it's going to chronically inflict damage on the biomolecules that make up our cells and tissues. Part of what goes on with oxidative stress is that you get generation of free radicals, which we call reactive oxygen species, or ROS. Some of the more damaging ones include superoxide, hydroxyl radical, and peroxyl radicals. Some free radicals are necessary in your body for signaling to take place. Most of these are not as necessary, and you may need some of these in very small quantities, but an overabundance of ROS is going to lead to damage. And they do tend to form as the result of stress, inflammation, infection, particularly. So bacterial infections can elicit in your body a high production of these free radicals. They target your biomolecules, your tissue proteins, lipids, and DNA, and they can cause damage. So where the antioxidants come in, there's several places where antioxidants may mitigate the damage caused by ROS. So some antioxidants kind of stop the formation of the free radicals before they have a chance to really get formed. So all of these processes that normally would produce a lot of reactive oxygen species, if we can downregulate some of the genes involved in these pathways, we can downregulate the production of these ROS. And so compounds that do that can be considered antioxidants. And then in a chemical way, a more sort of free radical reaction sense, once these reactive oxygen species are produced, we need to stop them from getting to their targets, right? So your biomolecules being the target, if you can ingest some compound, be it vitamin C, vitamin E, or a flavonoid, that will scavenge up free radicals, it's thought that this will reduce the amount getting to the target and causing damage. We also have defenses that work sort of in between the damage to repair the damage before it gets to the disease state. So before you actually get a full-blown cardiovascular disease, there may be repair processes at work in your body that could be boosted by the presence of antioxidants. So this kind of sums up what I just said. There's multiple roles of antioxidants in the body, some of which is chemical in nature, but a lot of it is about regulating or modulating biochemical pathways. From a chemistry point of view, what a polyphenolic compound can do is it can take the hit so that your body doesn't have to, right? So if you ingest Resveratrol, for example, and it has multiple phenolic groups, when it reacts with a free radical of any kind, it's going to generate what we call a phenoxyl radical, and because of resonance within this structure, aromatic compounds do tend to undergo resonance, it will gradually transform itself to, through this pathway here, what we call a quinone. So it may react once again and be oxidized a second time to form a quinone. And a quinone is a pretty stable structure. So this is just an example of sort of how these phenols absorb free radicals and turn into something that's more stable. And these compounds are better able to do that than your biomolecules that make up your cells and tissues. The wonderful sources of antioxidants. So it doesn't stop with cranberries and chocolate. We have green tea. Green tea contains a compound called EGCG, which is probably one of the most well-studied phytochemicals of the past 20 years. Lots and lots of research on EGCG. Lots of claims to fame. They even claim that it may help with weight loss. That might be a stretch. But I do think that EGCG has some potential to protect against cardiovascular disease and some cancers. Onions. Now, onions contain some of the same compounds that berries do, quercetin, which is one of my other favorite compounds. Onions have lots of it. Coming back to resveratrol, which was introduced by Susan, Resveratrol is actually produced by plants typically in response to stress, and it's not produced by all plants. It's produced by certain plants. It's especially likely to be produced in grapevines. When grapevines are infected with certain types of fungi, they will produce resveratrol. And when they make red wine, much of that resveratrol is preserved and concentrated, not as much in white wine. So the processing of white wine removes a lot of the resveratrol, unfortunately. So it's not quite as good for you, but it still tastes good. But red wine's a little bit more healthy. If you don't like red wine, you can still get resveratrol in things like peanuts, or you can actually buy it in a bottle, and you can actually buy a product called resveratrol juice that one of my students brought in for class one day. I also teach a class on food chemistry, and so we would have, sort of molecule of the week, and people would bring in food to go with these molecules, and somebody turns up with a big bottle of resveratrol juice, and it was just really funny. It's like, well, what does this taste like? It does taste kind of almost like a blueberry, sort of strawberry juice, but it's just a very strange concept. I think sometimes industry takes this a little bit too far. It's okay to stay with the natural juices and fruits. You don't have to actually concentrate it all into, one dose. But anyway, coming back to resveratrol, it is thought to contribute to the French paradox. So as a part of red wine, it may be providing some heart benefits. Studies have shown that it does decrease the oxidation of lipoproteins, which is one of the early steps in heart disease. Atherosclerosis and hardening of the arteries often starts with lipoprotein oxidation. Resveratrol has been shown to inhibit the growth of cancer cells and to possess anti-inflammatory activity. And more recently, some studies suggest that resveratrol is even able to extend lifespan by enhancing mitochondrial function. So lots of amazing things that this molecule may be able to do, and compounds like it. This particular molecule, it's a polyphenolic. It's not a flavonoid per se. It's a stilbene. So a stilbene, it's maybe hard to tell from this molecular model, but a stilbene is missing that sort of middle ring that you find in flavonoids. So a stilbene has two phenolic rings connected by a 2-carbon linker in the middle. So what do we actually do in the world of phytochemical research? So a lot of phytochemical research is on identification of new phytochemical nutrients. And at this point, phytochemical research has really taken off, so that in the past 10 years, a lot of the discovery has already been done. Lots of compounds have been identified. We may be looking for new food sources of these compounds, or better ways to get the food or the plant, the crop, to produce these compounds. We still do not know a whole lot about the mechanisms of action of some of these compounds. Some compounds are very well studied, resveratrol being one of them. Many are not. Many are not. We do need to identify the metabolites, because really what we want to know is what happens to these molecules when they get inside the human body. Really, because it's the metabolites that are produced by your body that are going to have that protective effect. So from a chemistry point of view, these first 4 bullets are sort of more chemically oriented. So this is the type of research that we focus on in my laboratory, but it's multidisciplinary work, so you really cannot do this without working in a team with biologists to assess the health benefits. And to assess health benefits, we typically use a series of different models, similar to the models that are used in the pharmaceutical industry to test drugs. We look at cellular models of disease, we look at animal models, and eventually, if there's enough money and interest, there may be clinical trials. So now on berries. My favorite topic. So Liz mentioned that cranberries is one of our most important crops in Massachusetts. We're actually the 2nd largest cranberry grower in the nation. We used to be the 1st, but Wisconsin outpaced us. They're just bigger. They can just grow bigger cranberry bogs. We can't keep up. But we make better berries. I firmly believe this. So cranberries belong to the Vaccinium genus. And as a matter of fact, so do blueberries. So the kind of cranberries that we have here in North America are Vaccinium macrocarpon. So macrocarpon is the species. Over in Europe, they have another type of cranberry called the Vaccinium oxycoccus. And it's somewhat similar to our cranberries. Traditional uses of cranberries included a treatment of urinary and bladder ailments, and this is where the whole UTI protection thing was born out of years of people taking cranberry products and drinking cranberry juice, and reduced incidence of urinary tract infection, particularly recurrent urinary tract infection. Blueberries, on the other hand, they are closely related. They're in the same genus. There are lots of different species of blueberry. These are just 3 of the most common ones. Low bush blueberry is the small wild blueberry that you find a lot of growing in Maine and Atlantic Canada. High bush, or the cultivated blueberry, the one you get in the grocery store most often. Those usually come from growing regions south of us, New Jersey and points south. And then in Europe and Scandinavia, we see a lot of bilberry, which is Vaccinium myrtillus. And so blueberry has some of the same compounds as cranberry because they're in the same genus, so a lot of the same biosynthetic pathways are triggered, but they're not identical. And so the traditional uses of these berries are a little bit different. Diabetes, many native peoples, particularly in Canada, use blueberry to help control blood sugar. Night vision, so blueberries have a protective effect on the eyes. And this was documented actually even back in World War II, where fighter pilots were given blueberry jam to improve their night vision. So as a chemist, what I'm interested in is to look at the relationship between the composition of cranberry and its biological effects. So the strategy is that we first take our fruit, and we work mainly with whole fruit, just so we don't miss anything. So we extract our fruit, and we get a fruit extract that contains literally hundreds of different compounds. And then in order to study these, we need to separate them. So we do a lot of separations in my laboratory. So we're not synthetic chemists. We really don't run reactions per se. Most of what we do is physically separate organic molecules based on their physical properties, based on their structure and their polarity. We can separate them. So we get groups of compounds called fractions. Fractions may or may not contain a lot of structurally similar compounds. We analyze their composition. So I'm an organic analytical chemist, so a lot of what we do is analysis, and we use a lot of the techniques that were introduced by Susan. And then once we have a good handle on what's in our sample, we want to see what it will do in models of disease. So here's where we team up with biologists, and we look at cell culture models of disease. Particularly in my group, we're interested in cancer and bacterial infections. So mainly we're looking at what these compounds can do in tumor cells or in a bacterial culture. Animal studies are also very important. So you want to see not only what does it do in cells, but once it gets into the body, what can it do? Animal studies being a little cheaper than clinical studies, that is usually the second step after you've established that there's some activity based on cell culture experiments. We also can use chemical tests to predict something about the properties and the composition of these extracts. So antioxidant and anti-inflammatory properties oftentimes can be determined using chemical testing. One of the things we always think about is whether there's synergy between the different compounds in the berries. So in other words, are we going to see a stronger effect with one compound or the whole mixture. And that oftentimes depends on what effects you're looking at. And in some cases, you see a stronger effect with an isolated compound. In other cases, you see a stronger effect with the whole extract. So these are some of the methods that we use. They're pretty simple methods. It's not a lot of cutting edge chemistry. There's no nano anything here. It's very basic, very simple, time-tested chemistry. To get your extracts, you're doing solid liquid extraction. It's just like brewing a cup of tea, basically. So we extract the fruit, usually by blending it and filtering. The chromatography, the separation, is this what you're looking at on the right, is a chromatography column in which we've applied a cranberry extract. And as you can see, cranberries are full of very colorful compounds, which makes it somewhat fun for students to separate until they've done it about 10 or 15 times. And they're like, I'm so done with this. But at first, it's like, wow, look at the pretty colors. This is great. I was like, yeah, that's why I like to work with these compounds, because they are interesting colors. And the color can aid us in the separation process because some of these groups of compounds are actually very differently colored. So we do our separations and then we do our analysis. So we usually start with an HPLC or a TLC analysis, which will help us to get an idea of how many compounds we have present or what type of compounds they might be. And then we go to more specific methods, which will help us pinpoint structure. So these are the methods that Susan mentioned, mass spec and NMR. And we do use a lot of mass spec. And then we have colorimetric assays, which can tell us a little bit more about the concentration of certain compounds within an extract. And can tell us a little bit about their antioxidant properties from a very simple point of view. Oh, scientists do love acronyms. Oh, and they just go on. And if you know NMR scientists, they've invented like a whole list of acronyms with really cute little names like cozy and nosy and stuff like that. And it really just means different ways to look at how atoms within a molecule interact. And how can we use that information to generate some info about the structure. And NMR is the #1 most important tool for structure determination. There is nothing better than that. With certain groups of compounds, if it's hard to isolate an individual compound, and I talked about packs, and that's a really difficult group of compounds to separate into individual packs. So we often study them as a group, and we use a technique called MALDI-TOF mass spec, which is matrix-assisted laser desorption ionization time of flight, which essentially means this is the way that the molecules are dispersed into the mass detector. And it's a milder sort of dispersion so that the molecules can get detected in one piece. They don't fragment as much. So it's a good technique to use for large biomolecules if you want to get an idea of the molecular weight. We use a lot of HPLC. So that HPLC is humming in our lab all day long. And that's going to give us a profile of the different compounds present in an extract. And for some things, TLC is a quick and easy way to look at composition. So you can quickly compare an extract with a standard and see if that standard turns up in the extract. So I'm going to start talking more specifically about some of our cranberry research. And on this slide are 3 of the compounds that we've been studying in cranberries that may have some beneficial health effects. How do we get these compounds? Well, as I mentioned, there's a lot of chromatography, right? And so we typically use what's called bioassay guided fractionation. And this is a strategy that's used by natural products scientists everywhere to break down a very complex mixture into smaller and smaller parts and then begin to pinpoint which ones may have a biological activity. So we can take our cranberry extracts now, and we've developed pretty effective ways to separate different groups of compounds. So if we're interested in the nonpolar compounds, we can extract those pretty easily and separate them using silica gel chromatography. and we can get our carotenoids, and we can get our triterpenoids, my favorite one, ursolic acid. And then if we want to look at the flavonoids or the polyphenolics, we're going to use a different procedure to extract and separate these guys and fractionate them into smaller groups so that eventually we have all our different groups of flavonoids separated. And then we can look at what they do. We can look at what they do as a combined extract, and we can look at what they do individually. So where we first started with cranberries was to look for cancer fighters. So basically, when you're looking for cancer fighters within natural products, you take your extracts or isolated compounds, and you test them in a panel of tumor cell lines to see if they kill tumor cells, basically. So our very first experiments with a panel of 9 different tumor cells suggested that, yes, some of the compounds in cranberry can kill tumor cells. What you're looking at here is the concentration data. So these are the dosages at which tumor cell growth is inhibited by 50% over the treatment period. How does this compare to Taxol? Holy God, if cranberries were as toxic as Taxol, we'd all be dead right now. So these compounds are not as toxic as Taxol, not by a long shot. But they do show some tumor inhibitory properties at reasonably high concentrations. Ursolic acid actually is a little bit cytotoxic. If you ate large quantities of it, it may cause some damage. But the quantity that we find in fruits, cranberries, apples, etc., is not going to kill us. In fact, it may be beneficial. The polyphenolic compounds are actually a lot less toxic to cells. In some regards, that's a good thing. All right? So there's different ways to kill a tumor cell. We can whack on it, like with Taxol, or we can chip away at it gradually using phytochemicals, right? Sort of a soft hammer approach. So these 3 compounds all showed some anti-tumor activity, different amounts in different cells. So some of these compounds may be better able to target certain types of cells than others. One of the ways that compounds can kill cells is by inducing what we call programmed cell death, or apoptosis. And apoptosis is a way that normal cells sort of maintain their good health, so that if a normal cell starts to have something go wrong, apoptosis will be triggered and the cell will die and it won't cause any further issues. But cancer cells, unfortunately, do not normally undergo apoptosis well. They found a way to navigate around it so that they can proliferate out of control and cause tumors to form and metastasis to occur. So if we can find a compound that will trigger apoptosis in relatively non-apoptotic cells, this is a good thing. And 2 of the compounds in cranberries do just that. This was an experiment in 2 different colon tumor cell lines. Actually, I think I only show 1 cell line here. But we looked at 2 different compounds. What we're looking for is a dose response. So as we increase the dose, are we seeing more cell death? And the answer is yes, we are. And which of these 2 compounds is more effective? Ursolic acid, really at high concentrations, causes a lot of cell death, which is why I say it is quite a bit more toxic than the polyphenolics, which cause a moderate amount of cell death. So they may be working by different mechanisms, and this is one of the things that we are now pursuing in my lab, is looking at how, in fact, do these compounds induce apoptosis, and what pathways, signaling pathways, are turned on or turned off in response to cranberry. Now, ursolic acid, yeah, it's a pretty good tumor killer. It's also a pretty good anti-inflammatory. And so this compound has been reported over the years in numerous medicinal plants. And when I very first started working on natural products, I worked with a colleague on medicinal plants from Peru, and we were trying to isolate compounds that had anti-cancer benefits. Ironically enough, one of the compounds that I first identified in my first project was ursolic acid. And lo and behold, it turns up again 10 years later in cranberries. It turns up in different forms, so some of these compounds may be esterified. So we have a carboxylic acid group here. You may have an esterification with this hydroxycinnamic group here, a phenolic compound. So, and these are two different isomers. We have the cis and trans, as Susan talked about. Both exist in nature, and they interconvert, which is kind of a pain when you're trying to identify structures. How did we identify this compound? We used NMR. We used a combination of proton and carbon NMR. Because it was a known compound, we just match up the data that we get with the chemical literature. There are databases you can search, but a lot of times you just have to go to the primary literature. And if you have some idea of what type of compound you're looking for, you start searching that group of compounds and looking at chemical data, chemical shift data, until you find a good match. Now, NMR, as Susan pointed out, gives you some idea of sort of the proton structure of a molecule. So what we're looking at here is an NMR spectrum of, down the bottom in blue, just ursolic acid. And this is just the 0 to 5 parts per million part of the chemical shift range. But each of these peaks does represent a proton in ursolic acid. We can also, if we have a high enough field instrument, get an NMR of the entire extract. And so we're working with some folks at Bruker Biospin to develop models to use NMR to predict from a given plant sample whether you in fact have cranberry or blueberry or some other plant entirely, to be able to predict what compounds are in there and how much. So ursolic acid, it's a little bit tricky compound to quantify using other methods. But we find that we can pretty easily quantify it using NMR because we have a few peaks that can easily be picked out of the whole big mess of peaks in the cranberry extract. And the peak area is proportional to the quantity, so we can use that information to determine how much ursolic acid we have in a given extract. So this is just an example of how NMR can be used not only to identify structure, but also to quantify compounds once you know what they are. Moving on to our flavonoids, we have flavonols. And flavonols are the three-ring structure. We have the polyphenolic functional groups. When we see flavonol, it tells us that there's a ketone in there, right? So here's your ketone. And in nature, this structure, quercetin, is fairly common among fruits and vegetables. It usually has a sugar attached. Some of these polyphenolic structures are not extremely water-soluble, so the plant makes them more water-soluble by tacking on a sugar or a series of sugars. There may be some small structural modifications in the ring structure here. For example, we have another flavonol, myricidin, which has an extra OH group here. This may or may not impact on its bioactivity. So you would have to do structure activity studies to see whether there is in fact a difference between quercetin and myricidin based on the presence of that one little OH group. Methylquercetin is just a methylated version of the original quercetin. So all of these are found in cranberry fruit, and the quercetin ones are found also in cranberry leaves. And they have lots of different sugars attached. So really, even within the group of flavonols, we have a very complex group of compounds. So if we want to study that, we need to get a profile of the different flavonols. For that purpose, we use HPLC analysis, high-performance liquid chromatography. And we can pretty easily detect quercetin and its relatives at 355 nanometers. Quercetin is a bright yellow compound, and it absorbs strongly at 355. So if we monitor 355, we see a series of peaks here. So HPLC is kind of like you put an extract into a sample loop and you flush it through a column with solvent. And as each compound travels through the column, they will travel at different rates depending on how well they interact with the column material. So some compounds will come out faster, some will take longer to come out. The ones that come out faster are these guys here, which are not flavonols at all. They are actually PACs. And they don't absorb at 355, but they absorb strongly at 280. So we can detect those guys too. But here comes this group of about 10 or 12 different compounds, and these are the flavonols, or quercetin glycosides. So each of these guys has a different, slightly different structural modification, most of which involves different sugars being attached. So we can determine the exact concentration of each flavonol, if that's important, or we can look at them as a group. Now, flavonols are antioxidants, and so one quick and easy way to predict if a compound is an antioxidant is to react it with a free radical. So we have an antioxidant assay that's a very simple free radical reaction. You can buy this DPPH radical, which is a hydrazine radical. It's a stable radical. If you keep it in the freezer, it lasts for quite a while, months in fact. And it's bright purple in color. It's very lovely. You make a solution of your DPPH, and then you react it with whatever you think might be an antioxidant. So it might be a solution, it might be a cranberry juice, it might be an individual compound. What we want to see is, does that compound quench the free radical? If it does, then violet color goes away, you form a yellow oxidized product, and you can monitor this change by monitoring absorbance at 517 nanometers. So it's a simple colorimetric assay. Now, early in the days of antioxidant research, this is what people did a lot of the time, are these simple colorimetric assays to see if, in fact, compounds can scavenge free radicals. Now we have a more complex understanding of antioxidants in that it's not just a free radical scavenging. We also have to look at how do these compounds interact with certain cellular pathways and mechanisms. So it has become a more complex field. But in food science, there's still a lot of simple antioxidant assays that are done just to compare samples in terms of how strong this one might be versus another one. So you may compare 20 different juices and say, oh, cranberry juice is obviously the best antioxidant based on this assay. And that may be true from a radical scavenging point of view, but some of the other ones may do other things. So it's just part of the picture. So finally, this last group of compounds here, the PACs, they're fun and interesting compounds to work with. There's a lot of structural variety, as I talked about earlier. So you may have anywhere from 2 to 12 or more of these 3-ring units linked together in these molecules. When they get in the body, most likely they're going to be broken down anyway. So there may not be a big difference between a 2-unit or a 5-unit molecule. We study these molecules primarily by using the mass spec method, MALDI-TOF mass spec, and what that can do is give us a profile of the different molecules based on their relative sizes, molecular weights. So the peak here at 997 represents a trimer. The tetramer, which was shown on the previous slide, there we've got 4 units, right? 1, 2, 3, 4 units of epicatechin. That would have a mass of 1285. Cesium is in there too, so it adds to the mass. That's where we find our tetramer. In a relative sense, mass spec can tell you kind of whether there's more of one compound than the other. It's not absolute. So we really don't use it for quantitative purposes, not this type of mass spec. We can also use a simple colorimetric assay to determine the overall concentration of PACs in a sample. So if we were comparing 2 different cranberry samples, or we were comparing cranberry to chocolate, which, by the way, also contains packs. We might use this assay to do a quick and easy quantification of the total packs. This assay uses the dimethylamino cinnamaldehyde reagent, and there's a condensation between the aldehyde and this, which is probably going to behave like an enol over here on this epicatechin ring, forms a green-colored adduct that you can pick out at 640 nanometers. So it's another simple colorimetric assay. I like this one a little bit less because it's not that reproducible, and I'm going to just come out and say that. But it's the industry standard. So people do use it in the food industry to do a quick and easy quantification or comparison of two samples, two pack sources. We need better methods than this one, unfortunately. Now, what do packs do? Well, some of the earliest studies about cranberries focused on urinary tract infection. So studies that came out in the 1990s began to look at how does cranberry prevent urinary tract infection? What are the sort of interactions between cranberry and the bacteria themselves and the urinary walls, the walls of the bladder? What was observed was that cranberry, when you drink cranberry juice or ingest cranberries, there's some process going on in your body. It may be a direct effect or an indirect effect, but the bacteria are finding it more and more difficult to bind to your epithelial cells. And if they can't bind to the cell, the wall of the urinary tract, then they can't establish an infection. So cranberries were found to prevent this binding process somehow. And this was called anti-adhesion effect. The actual molecular mechanism is still not well understood. However, there are many clinical studies that support the use of cranberry in preventing recurring UTI. It cannot cure UTI, so you cannot drink a gallon of cranberry juice and hope to get rid of an infection. Once the infection is established, your bacteria have a biofilm going, and it becomes very difficult to flush them out. So yeah, we still need antibiotics. So we need to think of functional foods like cranberry as more of a preventative or prophylactic. What else could berries protect against? Possibly stroke. How am I doing for time? 5 minutes. Okay, so I'm just going to briefly mention the stroke study. So I work with researchers at University of Prince Edward Island, one of whom is Marva Sweeney, and she's a cardiovascular researcher. She had done some studies on blueberry that suggest that blueberry can actually prevent brain cells from dying in the event of a stroke. We followed up the blueberry study with a cranberry study. Basically what's going on there is when you have a stroke, your brain is actually experiencing low blood sugar and low oxygen. And then when the blood flow returns, you're getting reactive oxygen species and further damage. So that sort of two-part process is going to cause a lot of cell death. We can use simple cellular models to predict whether compounds can inhibit this process. We can take those cells, treat them with cranberry or blueberry extract, expose them to stroke-like conditions, and then quantify the amount of cells that are killed. What we found is that cranberry does in fact inhibit this process, but it takes a lot of cranberry. All right? It has to be a very high concentration. So, okay, these are cells. Does it work in animals? Let me flip over to the, oh, there's my rat model. Well, we did a rat study. They loved cranberries. They ate lots and lots of cranberries for 6 weeks. Apparently, they were much happier than the control group that didn't get any cranberry at all. Were the results conclusive? Well, we thought there was an effect. This is the cranberry group. They look like they're not as damaged as the control group. However, it's not statistically significant, so it needs to be repeated with a larger group of animals. So this is always an issue with animal studies. However, we're fully confident that cranberries help in some way against oxidative stress effects in the brain. And a lot of Jim Joseph's research here at Tufts University has shown the same thing. Berries can help improve cognitive function. The antioxidants in berries are thought to actually strengthen the blood vessels and reduce the effects of oxidative stress. So lots of good things about berry. I know I'm running out of time, so I'm just going to flip through this real quick. Lutein. Cranberries have lutein too. It's good for the eyes. What do we need to do next? Well, we need to look at what's happening in vivo. So we have lots of cellular studies. We need to find out if, in fact, we get the same benefits in animals and humans that we do in cells. We need to know how these compounds are metabolized. We need to know how they work, so what are the mechanisms. And we need to know how much you need to eat to maintain health. And I'm getting these questions all the time, and I always say it's too early. We don't know. First, we have to show that there's an effect in humans, and then we can see if, in fact, how much you need to take, or if it's better to eat the whole fruit versus the juice. We have a UMass Cranberry Health Research Center now. We have a website set up. It gives you some information about cranberry research, particularly at the UMasses. So my parting advice is to eat your veggies and your fruit and have a Cosmo. But not before you drive. And I'd like to acknowledge all my students and collaborators that have worked with me over the years. And thank you for listening.
Yes, I wanted to know how stable are these phytochemicals? So when you cook cranberries and berries, do you lose a lot of the good stuff?
This is an excellent question. The answer is some of them are pretty stable, some of them are not very stable at all. Anthocyanidins, for example, those dark red compounds, and those are present in blueberries, by the way, too, are excellent antioxidants, which means that they themselves are easily oxidized. So if you store things in the heat, or you expose them to oxygen, or you cook them, those compounds are going to degrade. Some of the other compounds are a lot more stable. Ursolic acid, we can beat on it. It's very stable.
I just want to point out the fighter pilots and so forth in World War II that were given the blueberries. They were also given amphetamine.
Oh, nobody told me about that. That just kept them awake. The blueberries helped them see better.
Yeah, I have a question.
Yes.
You mentioned that the blueberries were used by native peoples to control blood sugar.
Right.
How do they know the blood sugar results?
Well, I think, from observation of behavior, most likely, but that early work of observation of the native peoples has been followed up by clinical studies going on in Canada. There's research at University of Quebec, in particular, looking at that.
So a lot of the phytochemicals that they promote, you hear, green tea is good for you, you hear chocolate is good for you, and then they come out with a study, tea is good for you, but not if you put milk in it. And chocolate is good for you, but only if it's dark chocolate, not with the dairy. So I'm wondering, is there a similar problem with cranberries and turkey?
Wow. Well, we definitely need to look at this.
I
Has a lot of
that seriously.
tryptophan. I don't know how that affects. It may be a question of how compounds are absorbed, right? So the tea thing, right? If you add milk or you don't add milk, that's going to affect how the compounds are absorbed in the gut. So I think for the tea question, that's probably a question of absorption. And so you won't know until those studies are done, right? And for the dark chocolate versus milk chocolate or white chocolate actually has no cocoa powder. And the compounds that are most beneficial are highly concentrated in cocoa powder. So as you increase the cocoa powder concentration, so your darker chocolates, your 65, 70, 80%, will be, much more plentiful catechins in there. And so by association ought to be healthier.
Okay, I mean, I know that's a kind of flippant question, but the question behind it really is that, you do these studies in the lab, but the actual practice, even the social customs of people, for instance, most people I know, well, they may drink cranberry juice, but if they don't, the only time they eat cranberries is at Thanksgiving. So there may be like social aspects to it, like I'll only drink tea with milk, and, so you lose that benefit. And I just wonder how quickly can you translate the lab results into how people should change their eating patterns?
Yeah, I think it's a long, involved process because you have to look not only at what's happening in the test tube, but you have to look at clinical trials and you have to look at epidemiological studies in order to determine, are these compounds beneficial? What dosages should people be taking? What form should they be taking them in? It's not so simple. Even within cranberry, there is conflicting clinical studies, some saying which there's a benefit, some say there's no benefit. Anecdotally, I will tell you that I believe there is a benefit from taking cranberry juice in preventing UTI, from personal experience. So,
Hi, I have a question about, so your area of research has to do with cranberries and phytochemicals in preventing disease and their role as antioxidants, preventing oxidative stress and preventing disease. Can you give a sense of what the relative impact of those phytochemicals and dietary factors compared to the other sources of disease like hereditary factors, genetic factors, or lifestyle factors like exercise or smoking or weight management, things like that? So many complex contributors to disease, and what's the relative importance of these different?
it's hard to say. I think you have to take everything together. I think that, people need to maintain a healthy lifestyle, whatever that may be. So that may include exercise and weight loss and high intake of fruits, vegetables, fiber, etc. Because we don't know the relative importance, of any of these things right now. And we don't know if it's going to overcome a hereditary disposition, say towards early breast cancer or something like that. It may help, but you may not be able to stop it.
Hi, I'm wondering, you have a lot of collaborators, and is the data from your work stored someplace together, and is it available to other researchers? How does all that?
So some of it's published. Some of it is, some of it isn't. So some of our cancer stuff is published, some of our stroke stuff is published. We're writing things up all the time, or trying to. We regularly present at conferences, so there may be abstracts out there giving a snapshot of what we're working on right now that may not be published for another couple of years when the study is completed. Some studies, generate data in a pretty quick way. Other studies take several years. So.
But do you archive the data someplace on a permanent basis?
In terms of raw data, no. We each have individual archives, so all the chemical data would be stored in my laboratory, and my collaborators would store all the biological data, and then we cooperate on manuscript preparation, et cetera, but we don't have a single archive. I could see where that would be a very useful thing, but we don't have that.
Is that generally true in chemistry?
Well, I think, it would depend, again, how much collaboration goes on. Some chemists exclusively work on the chemistry and don't work in multidisciplinary teams, so it may not be as essential for them, but I think, from my perspective, something like you described would be very useful. So my students would always have easy access to data that's not necessarily published yet.
I'd like to follow up on the, I'll pass it over in a sec. On the milk in tea, I think there were epidemiological studies out of England that showed a great decrease in esophageal cancer when milk was added to the tea, and this was stuff back from the 1940s.
Wow.
Now, this may not apply to some of the other teas that are much more in vogue now, But it is a point that I think is well established.
Well, that's interesting, and dairy actually has some health benefits due to some of the compounds, like there are different fatty acids, conjugated linoleic acids, and so forth, in dairy that may also confer a benefit. Yeah, perhaps.
So I just had, okay. When you go to buy cranberry juice at the store, I don't know if you've all been overwhelmed by the amazing choices,
Yeah.
cran raspberry, cran apple, cran blueberry. And I grew up on cranberry cocktail, which was loaded with sugar as well as,
Oh, yes.
cranberry juice. So I'm just curious in your studies, like your clinical studies of cranberries and helping prevent UTIs, if you use pure cranberry juice without sugar or because that's really, really, really tart, but.
If you've ever tried 100% cranberry juice, it's brutal. It's like drinking gasoline. The answer is that the clinical trials that are out there administered cranberry in different forms. So some of them use powders, some used juices, some used supplements, which are essentially powders. Those powders may or may not be made from just juice, or they may be made from whole fruit, but there's a variety of different preparations. And there was a movement by NIH when they were funding a lot of clinical trials, maybe about 5-6 years ago, to sort of standardize the products that were being used for clinical studies. And that didn't work so well, so I think now whatever is funded, you have to validate that, in fact, it is a cranberry product. You have to look at concentration. So with cranberry, it needs to be 27% cranberry. So if you have a cocktail, in order to see the UTI benefit, there's a certain dosage, and 27% cranberry, 10 ounces a day will give you that dosage. But if you're taking a supplement or something like that, and if you're taking it once a day versus twice a day, that may also change the effects, because there's the whole pharmacokinetics going on and how your body's metabolizing it. So it's a complex question.
Thank you.
That's it.
All right.
Thank you very much.
Thank you.